All right, I think we'll go ahead and get started. So this is Scott Baker on behalf of the Histocyte Society Board and our president, Mike Jordan. We'd like to welcome you to this session one of our two-part virtual annual meeting. So obviously a new endeavor for the Histocyte Society. And a list of our future dates for our meetings will be shared at the end of the presentation and can also be found on our website. Today's webinar is hosted by the Executive Board of the Histocyte Society, and there will be a survey at the end of the meeting that we would ask everyone to complete. Just a few housekeeping items for the meeting today. The participants are all muted, so in order to submit comments via the chat feature on your webinar panel, you can see on the diagram here below how to do that. Your chat will be seen by our staff and the panelists. Chat will be in controls at the bottom of your screen, and after clicking the chat window, it will appear on the right. Type your message and press enter to send it. If anybody wants to go ahead and give that a try and tell us where you're tuning in from, that'd be great. I can't actually see how many participants we have with my view, but it looks like it's quite a few. And if you have questions for the speakers, please put those in the Q&A box. So just to reiterate, there's a separate chat box as well as a Q&A box. But questions for the abstract presenters would be best put into the Q&A box. You can like a question in the Q&A box if your question's already been asked by someone else and you'd like to upvote it. And also just to note, a link to the meeting program will be dropped into the chat box. So if people have not received that and would like to download the actual meeting program and abstracts, they can do that. Due to time constraints, we may not be able to answer all questions. We have a very condensed schedule today, and we only have 2 or 3 minutes of questions after each abstract presentation. But then we do have some additional time for questions at the completion of the 3 abstracts that are being presented today. Just to note, the webinar is being recorded, and Histocyte Society members will be able to find the video in the members section of our website. If there's any technical issues you have, there is a Zoom help article that's in the chat window as well. We'd also today like to thank our sponsors for this year's virtual annual meeting, and those include the Histiocytosis Association and Cincinnati Children's Hospital. The Histocyte Society, just for those who don't know and who are not members, is a nonprofit group of over 200 physicians and scientists from around the globe. We're committed to advancing knowledge and improving outcome for patients with histiocytic disorders. And we have a focus on clinical and basic research and education. So for today's agenda, we'll start off with a presentation of the abstracts. At the completion of that, there will be a short question and answer period, as I mentioned, and then there'll be a business meeting for members. All non-members of the Histocyte Society will be asked to leave the call prior to the start of the business meeting. And you can see there are 4 reports that will be covered in today's business meeting. So we'll go ahead and get started with the abstracts. So our first abstract is being presented by Paul Milne, and it's entitled Mapping Non-BRAF Hematopoietic Clones in Multisystem LCH. Paul, we'll let you take it over.
Great. So yeah, thanks first of all to the organizers for the opportunity to present here online today. So this is all work that I did last year on a visit to Carl Allen's lab in Houston. Yeah, looking at trying to map the hematopoietic clones of multisystem LCH from non-BRAF V600E mutations. Next slide, please. Next slide, please. So, thank you. So we all know that up to 60% of patients with LCH carry the BRAF V600E mutation. A lot of work has been done on this particular mutation, with lots of groups now starting to personalize medicine based on detecting this mutation and tracking it. But we do know that a lot of other mutations are present in the MAP2K pathway, and there's variations in the genotype phenotype of the patients. We know the V600E mutation is associated with a resistance to therapy, an increased risk of recurrence, and more permanent long-term consequences. So this led us to question whether the cell of origin of LCH vary dependent on the mutation present. Next slide, please. So we've seen in our group that the V600E mutation is present in the peripheral blood mononuclear cells of patients, and it's predominantly in the myeloid cells, in the classical 14-positive monocytes, non-classical 16-positive monocytes, and the dendritic cell subtypes. And concordant results have been seen by several other groups now. Next slide, please. So we wanted to see whether the clonal architecture of V600E negative LCH varies from that of V600E positive LCH. So to do this, I fact sorted lots of the different subtypes of PBMC from the peripheral blood, amplified the whole genome using Repligy kits, and measured different mutations using droplet digital PCR. Slide, please. So for anyone who doesn't know, droplet digital PCR is simply lots of thousands of different PCR reactions happening within one experiment. Basically, the DNA is fractionated between thousands of droplets of oil, and each oil droplet has a PCR reaction running inside. So then you can get a PCR result from individual DNA strands. So on the bottom of this page here, you can see that you can either get results from one DNA measurement on the left, or you can get you can multiplex, so you can get graphs showing the percentages of wild-type DNA, which is green in this plot, mutant DNA, which is blue, and then droplets containing both mutant and wild-type DNA, which is orange. Next slide, please. So the inclusion criteria was fairly simple for this. It was patient samples, mostly from Carl Allen's cohort and a few from the Newcastle cohort, where a non-V600E mutation was known. PBMC samples were available from the freezers, and ddPCR primers were available direct from Bio-Rad, or I was able to design them myself. So there was a total of 18 patients enrolled, 6 patients with the BRAF indel, 1 patient with an alternate BRAF G466V mutation. 6 different MAP2K1 mutations were selected, 1 NRAS mutation and 1 ERBB3 mutation. We also had 8 BRAF V600E controls. Next slide, please. So it's been seen before that the stratification of patients, the presence of mutated DNA within the peripheral blood relates to the stratification of patients. Single system disease patients very rarely have any mutational DNA in the peripheral blood. Multisystem disease patients quite often have mutated DNA. In patients with risk organ disease, nearly all of these patients carry circulating DNA with the V600E mutation. Next slide, please. So looking at this cohort, using ddPCR to look at alternate mutations, we can see that pretty much the same pattern. No patient with single system disease had circulated mutated DNA. A fraction, about half of the patients with risk organ negative multisystem disease carried circulating mutated DNA, whereas the majority of patients with multisystem risk organ positive disease had circulated mutated DNA. Next slide, please. So the next thing to do is look into the specific cell subsets that carry the mutation, and I can go through the slide in great detail. This is the gating strategy I used to sort out 19 positive B cells, 3 positive T cells, the classical 14 positive monocytes and non-classical 16 positive monocytes, plasmacytoid DCs, and the CD1C positive DCs, which we now know from our recent paper in immunity can be split into 2 further subsets, the DC2, which is the true dendritic cell subset, and the CD163 positive DC3, which has a monocyte flavor. Next slide, please. So to do this, sorting cell subsets, and the amount of cells you can get from this is quite low, so I decided to amplify the DNA. So as part of the validation for this, I needed to show that amplifying DNA carrying these mutations was not biased in any way. So this is looking at lesional biopsy DNA from several of the patients and testing by ddPCR for their mutations before and after amplification. And you can see from the graph shown that there was no selective amplification or loss of sensitivity of the mutated or wild-type DNA for several of the mutations. Next slide, please. So first of all, just looking at some of the V600E controls, which were positive in the PBMC, they showed a similar pattern to what I've previously seen with the mutation present in the myeloid cells and mostly in the monocytes and DCs. Next slide, please. And then looking at the patients with the alternate mutations, a very similar pattern is shown with some very slight differences. The patients with the indel may possibly have lower levels of circulating mutated DNA. Patients with the MAP2K1 mutations, some of those patients had mutated DNA within the lymphoid fractions. These are very small differences. Next slide, please. So to conclude this work, we can see that the whole genome amplification and droplet digital PCR can be used for mutation testing in patients' PBMC and subsets. So we could use this to develop personalized medicine and mutation monitoring for a much greater number of LCH patients. There's a similar distribution of circulating mutated alleles according to the disease burden as has previously been seen in V600E. And the cell subsets circulating in the PBMC which carried the V600E, the non-V600E mutations, similar to that seen for the V600E mutation. Last slide, please. So with that, I'd just like to thank everyone from the Houston lab where I worked last year pre-lockdown and everyone in the Newcastle lab this year post-lockdown. All the patients and the clinicians for the samples and all the funders of the research. Thanks. Scott, you might need to unmute. Yeah.
There we go. Sorry, Paul, can you see the questions or you want me to read them?
Yeah, sorry, for the first question, trying to use ddPCR without amplification. I did try this initially and you get some good results from the cell subsets where there's a lot of cells like the T and B cells. But for the monocytes and especially for the dendritic cell subsets, there's just not enough sorted cells to successfully get the DNA into the oil droplets and get a valid result. And hi, Astrid from the Netherlands. And I did not include any MFP patients. So all of these samples are pre-treatment samples. So for the number of cells included, I wanted to get above 1,000 cells into the test, so I could at least be certain of a result of 0.1%. But I did test on some cell subsets which are lower than this. I just amplified with for cell sample types that I managed to get more than 1,000 cells. I still amplified just to keep the whole project consistent.
All right, thanks, Paul. Well, probably if there's additional questions that people have, you can continue to type them in the question and answer box, and we can cover those at the Q&A session at the end of this. But thanks a lot, Paul. We'll go on to the next abstract. All right, so our next abstract's being presented by Camille Bingenwald, and it's entitled BRAF-induced senescence drives Langerhans cell histiocytosis pathology or pathophysiology. So Camille, take it away and we'll watch for questions in the chat box.
Okay, hello everyone. Do you hear me well? Yes? Hello. So I do not have anything to disclose, and just to let you know that I made this work mainly in the laboratory of Dr. Miriam Merad at Mount Sinai. So next slide, please. So as Paul just said, As you know, the BRAF-V600E mutation is not only found in LCH tissue lesions, but also in CD34 positive hematopoietic progenitors, but at a very low rate, as you can see on this graph. And then this mutation is found mainly in myeloid cells, but not in T cells or B cells. So to be straightforward, the question we asked is how a BRAF-V600E mutated hematopoietic progenitor can give rise to a mutated mononuclear phagocyte. Next slide. So to address this question, we generated a mouse model where we enforced the BRAF-V600E mutation in hematopoietic stem cells using the SCL promoter. So we crossed those mice to Rosa YFP mice so we could track the presence of the BRAF-V600E mutation by the expression of GFP. Next slide. And so basically the BRAF-V600E animals developed an organomegaly, bone marrow aplasia, next slide, granuloma in the bones, and next slide, by immunohistochemistry, we could confirm the presence of CD207 positive myeloid cells in every tissues we examine, like the liver, the lung, and the dermis. Next slide. Then to confirm these results using human hematopoietic progenitors, we generated a model of humanized mice. So to be brief, we transplanted NSG mice with CD34 positive cord blood cells transduced with a BRAF-V600E lentiviral vector. Next slide. And indeed, those mice develop a very mild disease characterized by an accumulation of double positive CD1A, CD207 cells. So to conclude, the BRAF-V600E expression in multipotent progenitors is sufficient to drive Langerhans cell histiocytosis. So next slide. We took advantage of our mouse model to look specifically at the differentiation capacity of BRAF-V600E hematopoietic progenitors. And as you can see in red, strikingly, we observed a reduction of hematopoietic stem cells and multipotent progenitors in BRAF-V600E animals, while there was an expansion of granulocyte macrophage progenitor. Next slide. This GMP compartment was associated with an expansion of dendritic cell and macrophages and the bone marrow, but not neutrophils, suggesting that BRAF-V600E hematopoietic progenitors differentiate exclusively toward the mononuclear phagocyte lineage. Next slide. And we observed exactly the same when we kept into culture BRAF-V600E human hematopoietic progenitors that were transduced with a lentiviral vector. So next slide. In our mouse model, we have in the same animal a population of BRAF mutated hematopoietic progenitors and a population of BRAF wild type hematopoietic progenitors. So within the same animal, we can follow the differentiation capacity of BRAF mutated and non-mutated progenitors. And we observe strikingly that even the non-mutated hematopoietic progenitors were more prone to differentiate toward mononuclear phagocyte. Next slide. And we observe the same using our in vitro human culture. So basically, the cells that were not transduced with our BRAF-V600E lentiviral vector, but in close contact with BRAF-V600E positive cells, were also able to differentiate more toward the mononuclear phagocyte lineage. So this made us hypothesize that maybe the secretome from BRAF-V600E positive hematopoietic progenitors could participate in the myeloid skewing. And so to confirm this, next slide, we did something quite simple. We just kept into culture healthy CD34 positive cord blood cells in the supernatant from BRAF mutated cells or BRAF wild type cells. And indeed, when they were kept in BRAF mutated supernatant, the hematopoietic progenitors were more prone to differentiate toward the mononuclear phagocyte lineage. So we can conclude that the skewing of BRAF-V600E hematopoietic progenitors into the myeloid lineage is driven by cell intrasec, but also extrasec cues. Next slide. Yes, thank you. So as I mentioned earlier, we were very intrigued. So can you go back, please? Right. As I mentioned earlier, we were very intrigued by the fact that the BRAF V600E hematopoietic stem cells do not expand. And if they do not expand, we started to hypothesize that they might actually be senescent, as it has been described for nevi. So cells become senescent after a cellular stress like a DNA damage. And the cells, the senescent cells, are typically in an irreversible cell cycle arrest, but they are still highly metabolically active. So next slide. So we observed that BRAF V600E hematopoietic progenitors have a proliferative disadvantage compared to BRAF wild-type hematopoietic progenitors. So here I show you a bone marrow chimera competitive transplantation assay, and in red you can see that the BRAF mutated hematopoietic progenitors are not able to engraft when they are in competition with BRAF wild-type mutated BRAF wild-type progenitors. Next slide. And we confirm the absence of proliferation by the low BRDU incorporation of BRAF V600E hematopoietic progenitors. Next slide. In the tissue from our mouse model, we also observe that the LCH cells barely proliferate, as you can see by the low Ki67 expression. Next slide. Then we observe that our BRAF V600E hematopoietic progenitor cells in our mouse model express canonical markers of senescence. So high CDKN2 expression, high senescence-associated beta-galactosidase activity. Next slide. And we observe LCH cells, high CDKN2 and high senescence-associated beta-galactosidase activity. Next slide. And this was associated with a senescence-associated secretory phenotype, which means that senescent cells secrete a lot of cytokines, such as IL-1, alpha, beta, IL-6, IL-8. Next slide. And we observe this senescence-associated secretory phenotype in the bone marrow and at the tissue site. Next slide. So in human LCH lesions, we also observe a lack of proliferation as confirmed by the low Ki67 expression. Next slide. And we also noted that CD207 positive cells were also positive for CDKN2A in LCH tissues. Next slide. Next slide, and I will go back to this one, sorry. We also sorted specifically CD207 positive cells from LCH lesions, and we observed that they specifically stain for senescence-associated beta-galactosidase. And can you go back to the previous slide? So very importantly, in brain from neurodegenerative LCH patients, we observed the perivascular accumulation of myeloid cells that co-stain for CDKN2A. And this might suggest that senescence mononuclear phagocytes are recruited to the brain and might participate to the physiopathology of neurodegenerative LCH. So next slide, to summarize our findings so far, next slide. BRAF V600E hematopoietic progenitors become senescent secondary to DNA damage, but they are still able to differentiate and they accumulate in tissues where they secrete a lot of cytokines through the SASP. And we hypothesized that the SASP might contribute to the myeloid skewing of BRAF V600E progenitors. So it is well known in the senescence literature that the SASP production is driven by mTOR. So we use the mTOR inhibitor rapamycin to diminish the SASP. And you can see here, we treated cells in vitro that indeed the addition of rapamycin allow a reduction of IL-1 secretion. Next slide. And the secretion of the SASP allow a reduction of the myeloid skewing. Next slide. And when we treated our mice with rapamycin, we also observed in the bone marrow from our mice a reduction of the myeloid skewing. So this confirmed that the SASP participate in the myeloid skewing. Next slide. Finally, to confirm the central implication of senescence in LCH physiopathology, we aim to selectively eradicate senescent cells. As there is no drug that are absolutely selective for senescent cells, we use another mouse model. So to be brief, we crossed our previous mouse model that I already introduced you with ink attack mice. So in ink attack mice, senescent cells that are CDK and 2A positive can be visualized by GFP expression and can be deleted upon administration of a drug. Next slide. And indeed, in this new mouse model, we observe a specific accumulation of GFP positive senescent cells in the bone marrow, but also in the lung. And these cells were deleted after the drug administration. Next slide. The GFP slides, whatever the organs observed, were mainly myeloid cells. Next slide. And moreover, when we deleted the senescent cells through the administration of the drug, we observe an improvement of the outcome of the mice. So indeed, we observe a reduction of the organomegaly. Next slide. A reduction of the tissue infiltration and a reduction of CD207 positive cells. So next slide, this is my last slide. So to summarize a bit our study, BRAF V600E progenitors start to become senescent in the bone marrow after DNA damage. The SASP they secrete participates partially in the myeloid skewing of mutated and non-mutated progenitors. Then these senescence hematopoietic progenitors are able to differentiate toward mononuclear phagocyte lineage, and they migrate into tissues where they still secrete the SASP, which is partly responsible of tissue destruction and recruitment of immune cells, leading to granuloma. So we believe that eradicating the SASP through mTOR inhibition or eradicating senescent cells through senolytics can be an alternative of treatment in LCH patients. Next slide. So I really would like to thank my mentor, Miriam Merad, and also quickly, I would like to thank Carl Allen and Jennifer Pikarsik from Cincinnati's hospital. Thank you.
All right, we probably just have time for maybe one question.
So there is a question. Did you look at what happens with BRAF inhibitor treatment in comparison to RAPA on this SASP phenotype? No, we use control or rapamycin. We didn't use BRAF inhibitor to look at the SASP.
All right, thank you, Camille. If there's other questions, go ahead and type them in the Q&A session, and we'll come back to this during that session at the end of the next abstract. So, but we will now move on to our third and final abstract being presented today by Jessica Velasquez, and the title of this is T-cell receptor sequencing in patients with Langerhans cell histiocytosis. So Jessica, you can take over.
Okay. Can everyone hear me? Yes. Okay. So hi everyone. I would like to thank the organizers for this opportunity to speak. I am part of Dr. Carl Allen's lab, and I will be presenting our work on characterization of distinct T-cell receptor repertoires in patients with LCH. Next slide, please. So I have nothing to disclose. Next slide. So as everyone is familiar with, LCH has variable clinical presentations, but despite that, all LCH lesions look alike histologically. They have a high presence of CD1A positive, CD207 positive pathologic dendritic cells. And although clinical outcomes have improved over the past decades, there is a need for better therapeutic options. Next slide. Next slide, please. So here are two different models of immune response. In healthy skin, epidermal Langerhans cells, which are antigen-presenting cells, are able to activate, oh, can you go back, please? In healthy skin, epidermal Langerhans cells, which are antigen-presenting cells, they are able to activate T cells in an antigen-dependent manner. In various cancers, we see there is a tumor-mediated immune response in which antigen-presenting cells are able to present tumor-derived antigens. LCH is quite unique because the pathologic cells themselves are antigen-presenting cells. We want to understand if and how the pathologic cells interact with T cells. Next slide. So as we know, LCH lesions are comprised of a large inflammatory infiltrate. We have identified T cells to be the most prominent of these immune cells, apart from LCs. And among these cells, we found CD4 T cells to be more numerous than effector CD8 T cells. And as previous studies have shown, we also saw an accumulation of regulatory T cells. So there is a large presence of T cells in LCH lesions, but their impact on LCH pathogenesis is not well understood. Next slide. Therefore, to better understand the role of these T cells, we perform TCR sequencing and RNA sequencing on a cohort of patients. With TCR sequencing, we hope to be able to determine whether a subset of T cells have undergone clonal expansion, which could be elicited by a particular antigen. So I'd like to review what occurs in T cell receptors in healthy and disease conditions. So under healthy conditions, the immune system is unaffected by antigen stimulation. We see random rearrangement of the TRBV and TRBJ gene segments, and T cells show a polyclonal state. However, under disease conditions, the immune system could be stimulated by a prospective antigen, which could lead to the selective enrichment of certain gene segments. Looking deeper at the interaction in T cell receptors, we see that the CDR3 region is the most highly variable region on the T cell receptor. So it can act as a tag for T cell clones. Next slide. So here's our study design. We obtained lesion and matching peripheral blood from 9 LCH patients, blood samples, and then we sorted for helper CD4 T cells, cytotoxic CD8 T cells, and regulatory T cells, and then performed alpha beta TCR sequencing and bulk RNA sequencing. And this table to the right summarizes the LCH patients included in our study. Next slide, please. Now to look into our results. So first, to evaluate TCR repertoire diversity and clonal expansion, we assess 2 things: the total number of unique CDR3 amino acid sequences and Shannon's diversity index, comparing LCH lesion to control tissue. The benefit of Shannon's diversity index is that it normalizes the number of unique clonotypes to the total number of sequences. So a higher index value indicates a greater diversity of clones that are more evenly distributed in frequency, while a lower index value indicates a lower diversity of clones with specific clones dominating in frequency. Next slide. So in all 3 LCH lesion T cell subsets, we found fewer unique CDR3 clonotypes and a lower diversity. This could indicate that specific T cell clones are undergoing clonal expansion, suggesting that a potential antigen-involved DC T cell interaction could be present. Next slide. Next, we wanted to determine if any CDR3 sequences were shared among patients. So I'd like to quickly orient ourselves to these circus plots. So L1 lesion refers to the lesion sample of patient 1, L3 lesion refers to the lesion sample of patient 3, and so forth. So we had 9 patients, so we will see L1 through L9. So each chord represents a shared sequence between 2 patients at 100% similarity, and the width of the chord represents the frequency of that shared sequence. So here we see that out of 9 patients, 4 share CDR3 clonotypes in the lesion CD4 T cells. Next slide, please. So we saw shared clonotypes in all 3 lesion T cell subsets. While the CD4 group had 4 patients with shared clonotypes, the other 2 groups only had 2 patients each. Next, please. We next wanted to determine if any clonotypes were found shared in the lesion and blood of these selected patients. And we in fact did see that. And these clonotypes are indicated by the red arrows. So as you can see, the lesion and blood of patient 1 share clonotype in CD4 group, and the lesion and blood of patient 1 share clonotype in the CD8 group. and the lesion and blood of patient 9 shared clonotype and the regulatory T cells. Next slide, please. We also found shared clonotypes among the blood of, oh, can you go back, sorry. We also found shared clonotypes among the blood of multiple patients, just as we saw in the lesion. And these clonotypes are indicated by the black arrows. You can see that the blood of patient 1 shares a clonotype with the blood of patient 7 in the CD4 group, and the blood of patient 1 and 4 share a clonotype in the CD8 group. Next slide, please. So since we observed shared CDR3 sequences among selected patients, we performed conserved amino acid motif analysis to identify sequences with 90% similarity among all the patients. The top conserved motifs that we found for each T cell subset and sample type are represented by these sequence logos. They suggest that similar or same epitopes are observed among LCH patients, leading to the possibility of a common antigen. Next slide, please. Next, we assess the frequency of each TRBV gene segment among the lesion and blood of our patients. And this is represented by the heat map shown here. We found that several V beta gene segments shared a high frequency in both the lesion and blood of multiple patients. And these gene segments are indicated by the red asterisks. Next, please. So interestingly, this trend was found in quite a few segments of the TRBV7 family in all 3 T cell subsets, as shown in these tables to the right. So here we list the patients and sample type in which we observe high frequency of this specific family. This is quite interesting and something we could look further into as the selective enrichment of TRBV7 could be formed from the stimulation of an antigen, suggesting that there is a potential involvement of antigen selection. Next slide, please. So to conclude, our data suggests that a potential antigen-involved DC T-cell interaction could be responsible for the clonal expansion observed in the LCH lesion T cells. As similar CDR3 epitopes were observed among LCH patients, there is a possibility of a common antigen in LCH. And the selective usage of TRBV7 in LCH patients could be molded from their interaction with a potential common antigen. So our future goals include trying to characterize this potential antigen and determining if the same TRBV clones could be found in repeated biopsies of multiple patients. So there's still more to understand, but we believe this has broadened our view on the interaction between T cells and LCH lesions. Next slide, please. So I'd like to thank Rikia, Dr. Allen, Dr. McLean, and all of our lab members and our funding sources. And thank you again for letting me present our work.
Great, thank you, Jessica. If there's any questions for Jessica, please type them in. Actually, scroll down here. Jessica, can you see the Q&A box, or I think there's one from Astrid.
Yes. So can you speculate whether the CD4, CD8, and regulatory T cells shared in CDR3 clonotypes and the V beta gene segments may be primed? Could you explain your, sorry, I don't understand the question really.
So maybe Astrid could, Astrid would probably have to type a new question, I would guess. Maybe try to rephrase that or add to it, and we'll come back to it. And we could skip to the next next question there, Jessica.
Let's see. Do you think that it's the actual, forward. Oh. Isn't, okay, I see. Do you think that it's the actual LCH cells themselves responsible for antigen presentation? We don't know for sure if it's the actual pathologic cells responsible for antigen presentation, but that is something we are working into looking at.
Hey, Jessica, this is Mike Jordan. Can I ask you a question? I can't type it into the box. I'm wondering if this antigen may be self-antigens, and I think this is sort of the same question maybe Stefan Ladish is asking as well. Or do you think that this is in fact perhaps a self-antigen that the T cells are responding to? Did you hear my question, Jessica?
So if this is in fact a self-antigen, yes.
Yeah, or what the antigen is at all.
So right now, we're trying to see if this, the epitopes that we found among the LCH patients, if they could be found in other studies. So we're trying to input the sequences in databases to see if they're found in other studies. But the limitation on that is that most of the studies are virus-based. So most of the sequences that are epitopes that are coming up are have to do with viruses.
Okay, great. All right, thank you very much.